Good morning. I'm really excited to see so many people here this morning. It's great to have you here and be able to tell you about our work, but also thank you for the work that you do, which is, the important part. You guys we're standing on the shoulders of giants, and you guys are the giants. You make this work possible. So as Dr. Buckner said, can you hear me okay? Okay. As Dr. Buckner said, my interest is in genetics, and I have a number of different projects working on different autoimmune diseases, but one of the areas I'm going to talk to you about today is systemic lupus erythematosus, or SLE, and specifically some of the genetic underpinnings that have been uncovered in the last few years and how that's pointed us towards new therapies. Okay, so I'm going to begin by telling you a little bit about our Hermetic disease registry, because Dr. Buckner alluded to it first, but I wanted to give you some more details. So this is our Hermetic disease repository and registry, and it's made up of 4 different diseases. So we have lupus. We have about almost 400 patients who have registered in our repository and donated samples. Rheumatoid arthritis with 482, and not shown on this slide is that we actually have a registry of first-degree relatives of RA patients as well. So that's really important in terms of understanding what are the predisposing factors, what are the symptoms that start appearing first. So we learn a lot of important information from that. We have a scleroderma registry of 242 participants, and then relapsing polychondritis with almost 300. And the principal investigator for our rheumatic disease registry is Dr. Carlin across the street, but Dr. Buckner is also a co-PI, and the clinical coordinator, the person who you may interact with, is Sylvia Poso. You may also have interacted with Melissa Pita, which we sadly lost her to the Hutch recently, and then some of our other coordinators are working with Sylvia to support the rheumatic disease registry. So the registry started with Dr. Buckner and relapsing polychondritis, and that was in the late 90s. And then in the early 2000s, we added these other diseases, and specifically lupus was added as part of funds that we received from the Washington State Life Science Discovery Fund, which is where the tobacco money, the funding that came from when the tobacco settlement was made. So that was turned into research funds, and so that's how we funded the start of our lupus registry. So think of that as a good outcome there. And we have given, I'm sure this is an underestimate, of over 300 samples to other investigators and collaborators. Okay, so what is lupus for those of you who might not have it? So lupus, like other autoimmune diseases, is a systemic disorder. So it affects many places in the body, and those are sort of shown over here. You could have lung problems with pleural effusions, heart problems with arterial sclerosis. One of the big complications with SLE is lupus nephritis, where it affects the kidneys. Anemia, arthritis, so joint problems, Raynaud's phenomenon, which is where you get the blanching of your fingers. It can affect your skin. As you can see here, the classic butterfly rash. There's photosensitivity, and there can even be neurologic problems. And some of the features of lupus is that it leads to long-term inflammation, so it's chronic. And the prevalence across the world is between 20 to 150 people per 100,000. And sort of the classic lupus develops between the ages of 15 and 45. Now, it does affect children. There is a childhood lupus, which tends to be more severe, and then it can affect people beyond the age of 45. In particular, men tend to be affected more later. Now, lupus within this time frame affects women more frequently than men. There's still many men who have lupus. and it's more frequent in women of African American, Asian, Latino, or Native American descent. And what I'm going to tell you about is it actually involves both the innate and the adaptive immune system. And you've heard about this already this morning from Dr. Lord and Dr. Kita. So these are the players in lupus. So you have your innate immune system. These are the cells that are always poised, always ready, sort of your first line of defense. In lupus, there's sort of three players. There's these plasmacytoid dendritic cells. These are, in lupus, the bad guys because they secrete a lot of type 1 interferon, which is one of the major inflammatory hormones in lupus. There are myeloid dendritic cells. They can also secrete inflammatory cytokines. You see here TNF, and you just heard about that from Dr. Lord, tumor necrosis factor. And then you have the macrophages. They're the scavengers. They, in a normal immune reaction, they would go out and eat the bacteria or the viruses. And they also secrete inflammatory cytokines, but also, importantly, in lupus, this growth factor that makes B cells grow. And we're going to talk a lot about B cells today, and that's called BAFF. Now here's your adaptive immune system. This is the portion of your immune system that remembers that when you have a vaccination, it remembers later when you see that particular organism again. So you have your T cells. You've heard about those this morning. Two flavors, the CD8 T cell and CD4 T cells. They can also secrete these inflammatory hormones as well. And then we have our B cells. This is the world, my world is very B cell centric. B cells are the immune cells that make antibodies. You've heard about them. Those are the circulating molecules that normally go out and inactivate bacteria and viruses. But in lupus, we get antibodies that actually bind to your normal cellular products, and then these complexes of these antibodies clog up your organs. And I think that's shown in the next slide. So what are the factors that can lead to the development of lupus? Well, there's many. There's genetic, which I'm going to talk to you about today. So there, in the last 10 to 15 years, there's been a lot of studies of lupus patients compared to control subjects who don't have lupus, looking at what are genetic variants that are found in the DNA that are more common in people who have lupus than in healthy controls. And we found a lot of different variants, like 40 to 50 different variants. So lupus is what we call a complex genetic disorder. It's made up of a lot of different genetic variation. They aren't big, bad genetic variation. They each give a little bit. It's like going to a buffet at King's Table or something. You know, everybody has a little bit, but nobody eats the entire buffet. And we think that that may determine what kind of lupus you get. You've heard this morning that autoimmune diseases can be very heterogeneous, that not everybody is exactly the same. And part of that may be because different people have a different combination of genetic variants in their background. Now, just having a genetic variant by itself doesn't mean you're going to get lupus. It takes other things. So environmental factors. And some of the environmental factors that have been linked to the development of lupus are UV light. Now you would think, since we're in the Northwest, that wouldn't be an issue. Viruses, smoking. Smoking is also a huge risk factor for rheumatoid arthritis. And then as Dr. Lord was just telling us about the microbiome, we're learning more and more in the last 5 years about how the bacteria in our guts help to shape our immune system. And it's a good thing, usually, because they direct and sort of teach our immune cells to behave. But we are starting to find that with autoimmune diseases and other diseases, including obesity and things like that the microbiome can differ. And we don't know if that is the chicken or the egg yet, but we're learning more about it. This is going to keep your ears open because you're going to hear more and more about this. Then hormones play a big role. this is particularly true for lupus in women. The female hormones, pregnancy can bring on lupus. And then why is this? Well, we know one thing is that some genes that are associated with lupus are found on the X chromosome, which women have 2 copies of the X chromosome and men only have 1. So they have more of these genes. And then there are what we call epigenetic modifications. So the genetic variants are actual changes in the spelling of your genes. Epigenetic changes are when the DNA itself gets modified and it affects the expression of these genes. And one that I'm going to talk about later is IL-2. Okay, so how does the immune response cause end organ damage? So the major players for lupus are these inflammatory cytokines that the different immune cells are secreting, and importantly, the antibodies, because in lupus, these antibodies tend to be against cellular material. And why is that? Well, we have dying cells. They're called apoptotic cells. And little blebs of those come off. They have DNA in them, they have RNA in them, they have pieces of the proteins that coat the DNA, and that's what these autoantibodies recognize. So then you get complexes of the pieces of cell plus the antibody, and then these are the damaging things. They can make the innate cells secrete more of those inflammatory cytokines, more type 1 interferon, and then the complexes of the autoantibody and the pieces of cells can clog up your organs. So here's an example of a glomeruli from a kidney, and you can see the antibody complexes here. And then, like I said, the antibody complex can bind to a monocyte and secrete more cytokines. Here's an artery where it's causing an arterial plaque. And then also in neurons, these autoantibodies can bind protein on the neurons, leading to neurologic developments. So what about the genetic risk for lupus? Well, we know that most autoimmune diseases have at least some genetic basis, and that's shown in this slide here. So you can see type 1 diabetes, Graves' disease, multiple sclerosis, and here's lupus and rheumatoid arthritis. And what this is comparing the risk of a sibling to someone who has an autoimmune disease to the risk of someone from the general population. So this relative risk, this is how much higher your risk is of developing an autoimmune disease if you have a sibling compared to the general population. And what you can see is that for lupus, you're 20 times more likely to develop lupus if you have a sibling who has lupus. And the same for multiple sclerosis. Those are kind of the worst. Type 1 diabetes and Graves' disease, 15, RA, 8. So lupus has a big genetic component. And as I was telling you, the last 15 years, we've started to understand what these genetic variants are. And so that's kind of shown on this slide. So you'll see the different immune molecules again. So the first take-home point is that almost all of the variants that are associated with the development of lupus are found in immune-related genes. So we were right, lupus is an autoimmune disease. These genetic variants prove that. And then they point to the cells that I was just telling you about. So there are a lot of genetic variants that affect B cells. So see this whole list, that's why I'm very interested in B cells. There are some genetic variants that affect T cells. And the innate cells that show the T cells, the pieces of the cell that make the B cells make the autoantibodies. Then these B cells develop to the certain kind of B cell called a plasma cell that secretes the autoantibodies. And then the autoantibodies bind to their piece of cell, and then normally they would be cleared. But there's a whole series of genes in lupus that actually impact the clearance of these autoantibody complexes, and they also affect the clearance of the dead cells, the apoptotic cells. So you're not, once these things form, once the cells die, they're not getting removed quite as well. And so that's leading to a predisposition to lupus. And then you have your inflammatory cytokines, and we know in particular for type 1 interferon that's so important in lupus that there are a lot of genes that impact the type 1 interferon pathway. So the genes are directing us to pathways that are important to target in lupus. And the other thing we learn about genes, and it's not specifically depicted in this slide, is that we see some of the same genes over again from autoimmune disease to autoimmune disease. So one gene in particular I'll mention, PTPN22. Dr. Buckner has done a lot of work on this gene in conjunction with Dr. David Rawlings at Seattle Children's Research Institute. PTPN2 is associated with rheumatoid arthritis, lupus, type 1 diabetes. It's a bad deal. So that's the other piece that genetics tell us, is that there are common mechanisms to autoimmune disease, and those are particular areas that are useful to target. So as I said, I'm really interested in B cells, and there's a long list of genetic variants that are associated with B cells in lupus. So B cells, as I said, they make the autoantibodies which bind to the pieces of the dying cells. These immune complexes then cause the innate cells to secrete inflammatory cytokines, and they get clogged in your kidneys and can cause damage to the neurons, arterial plaques, etc. So, and then B cells can also show the pieces of the dead and dying cells to T cells, and then you get your autoreactive T cells inflamed and kind of all revved up. And then on top of that, the B cells themselves can secrete these hormones, these cytokines, and those cytokines can act on the T cells, they can act on the innate cells. So B cells are very important in lupus, and the fact that there are so many genetic variants that affect B cells and the development of B cells points to that. It's saying, here, look at these guys are really bad, you should think about targeting them. So we have these B cell genes, so this PRDM1 is really important in the cells that secrete the autoantibodies. Then we have several genes here which affect how the B cells get activated. And then a gene here, TLR7, this is thought to be possibly important in lupus that affects men, is a gene that makes the B cell very activated, but also innate cells. And then lastly, I'm going to talk a little bit about BANK1. BANK1 was associated with lupus several years ago, and it's not really known how BANK1 functions and how it normally functions, but how its function is altered in lupus. And so that's one of the projects that I've been working on in collaboration with Dr. Buckner and Dr. Rawlings and Dr. James at Children's Hospital. And this is just a piece of our data. So we study healthy controls to help sort out what the impact of a genetic variant is. Well, why would we do that? Why would we study healthy controls? Well, in patients with lupus, they have a lot going on. They've got inflammatory cytokines, they've got other genetic variants. They may be on therapy, especially for like lupus and RA. So we find when we look at genetic variants in control subjects, we can kind of sort out all that, and we can just kind of hone in on the effect of this genetic variant. And so that's what we're doing here in this graph, and we're looking at the frequency of B cells. And what you can see is that when we look at controls that don't have the variant in the BANK1 gene, they have a low number of B cells. And when we look at healthy controls who happen to carry the version of the BANK1 gene that's associated with lupus, they have a higher number of B cells. Now what's important is that when we look in healthy controls compared to lupus patients, we also see an increase in B cells. So what this is telling us is that the BANK1 variant that's associated with lupus may be one of the mechanisms by which patients with lupus develop high numbers of B cells. So this is how we are applying this genetic knowledge. It's also helping us to develop new targets in lupus. So if you have lupus, you know that most of the therapies have been aimed at tamping down the immune response with steroids. But we're starting to get biologic agents and other small molecule inhibitors that are going to be applied to lupus. So this is a very exciting time for lupus. And what you can see is that a lot of these new drugs are aimed at the players that we've talked about in the immune system. So here's your B cell. Here's the type of B cell that makes the autoantibody. Here's your immune complex. It's going to clog up your kidneys. Here's your antigen, your apoptotic cell. Here's your innate immune cell. Here's your T cells. So one of the first drugs that was developed for a variety of different autoimmune but also B cell cancers was this drug rituximab, which is known as Rituxan. And this is going to deplete your B cells. Now, sadly, Rituxan was not that effective. And surprisingly, it was not that effective for lupus. One reason might be that the cells that are making the autoantibodies don't express the molecule CD20 that Rituxan targets. So it's getting rid of all these cells, but it's not getting rid of these cells. And they're long-lived. They live in the bone marrow, and they can come out and make more autoantibodies. So that might be one reason why Rituxan has not been as successful as hoped. But here's a new therapy, Benlysta or belimumab, which is targeted at this B cell growth factor. Remember I told you that the macrophages make this B cell growth factor? It's going to make those B cells, so if you already have a propensity to have more B cells, it's going to make them grow like crazy. It's like putting Miracle-Gro on your plants in the garden. So Benlysta binds to the BAF molecule, the B cell growth factor, and inactivates it. So this is a new drug that in clinical trials appeared to be helpful, and it's not in all patients, it's in some patients. And that's one thing that genetics can do. They can help us to stratify patients to figure out who is going to be more likely to respond to a particular therapy. Here's another drug, anti-IL-6. This is currently being used in rheumatoid arthritis with great success. And I have to write down the common names of these because I am not a medical doctor. Actemra. Infliximab or Remicade is the anti-TNF. Humira is another anti-TNF agent. And over here we have abatacept or Orencia, which is going to block the interaction between T cells and B cells, and also the interaction between T cells and these innate cells, and sort of prevent the activation of T cells and B cells. A new drug that's just coming out, tofacitinib. See, if you can pronounce these names, they let you work here. That's actually a small molecule inhibitor, which is kind of interesting because you're much more likely, less likely to develop an immune response against the drug itself. And it actually inhibits the enzymes that are doing the signaling within the cells. So that's a new drug that has been developed and is being applied to autoimmune diseases. And then the last thing that I want to talk about is a drug that is very early in its infancy. It's just been tried in 1 or 2 patients, but it's showing surprising progress and promise for lupus, and that is low-dose IL-2. So IL-2 is a growth factor for T cells. It's sort of the equivalent of BAF. And this was a surprise to me, but it was very effective at ameliorating lupus in the few subjects that it's been tried in. And I was like, well, why would that work? Well, you heard Dr. Lord talk about regulatory T cells. These are T cells that put the brakes on the immune response. And the IL-2 gene is a gene that is turned off in lupus. It's epigenetically modified. So lupus patients tend to have low levels of IL-2. And as a result, they don't have a lot of regulatory T cells, or they have lower numbers of regulatory T cells. But so by giving a very low dose of IL-2, you can specifically enhance the development of the regulatory T cells without turning on all the T cells, because the regulatory T cells are very sensitive to IL-2. So I'm just going to show you a snippet of data. This is one patient. This is in press in the Annals of Rheumatic Diseases. This is one patient who was given 4 rounds of IL-2. Those are shown by the gray bars here. Now this is looking at their lupus severity. So this is time. These were the days after treatment. And this is the lupus severity. And what you can see is that when they were given IL-2, the lupus severity went down. Here's a picture. You can see that the rash on the legs is alleviated. And then this is what happens to the regulatory T cells. So every time they get IL-2, the regulatory T cells go up. And then you can see afterwards that when they weren't given IL-2, the regulatory T cell numbers went down, stayed down, and their lupus severity started to come up again. So this is really exciting. Look for it. This is going to have to go through clinical trials, but I think it's a new and exciting pathway in lupus. So just to recap, I told you about that lupus is a systemic chronic autoimmune disease, and two of the major players in lupus are autoantibodies, which are made by Bcells, and cytokines, which are made by both innate and B and T cells. And there are many factors that contribute to the development of lupus: genetics, environment, hormones, to name a few. And there are new studies that have identified some of the genetic risk factors that can lead to lupus, and those fall in immune genes. They fall in pathways that indicate that they are affecting B cells, T cells, cytokines, and clearance pathways. Some of these are shared with other autoimmune diseases, and they target us to look at these areas for developing new therapeutics. And here at BRI, we're very interested in linking these genetic variants in B cells with alterations and trying to understand how does lupus develop, how can we target it. So I'm going to stop there. And the last thing I just want to say is that we could not do this work without you, and I'm very appreciative and honored that you are willing to participate in our research and to advance work that is going to help patients with lupus, both here and at other institutions that we share our samples with. So you are the real drivers of this work. And I'm going to ask Dr. Buckner to come up because, as I said, in the New Yorker cartoon, I am not an actual doctor, I'm merely a PhD.
Well, let's thank all the speakers and then we'll